Okay, let's talk about cancer. We all think we know the main culprit, right? Bad genes. But what if I told you there's something else? A kind of ghost in the machine, pulling the strings. That's what we're diving into today. Some absolutely fascinating new research that's flipping our understanding of this disease on its head.
Yeah, for decades, the story has been pretty straightforward. Cancer is a disease of the genome. You get mutations in your DNA, and boom, cells start growing out of control. Simple, right? But what if that's not the whole picture? What if it's only half the story? It's starting to look like these genetic mutations aren't acting alone. Nope. It seems they've got a hidden accomplice, a silent partner, working in the shadows to really push cancer forward.
And this silent partner, it has a name: DNA methylation. Now, stick with me here. Think of your DNA as the actual hardware in a computer, right? The physical circuits and chips. Methylation doesn't change that hardware. It doesn't rewrite the fundamental code. Instead, it's like the software running on top. It slaps these tiny chemical tags onto the DNA that basically act as on-off switches, telling your genes when to power up and when to shut down. This whole idea is the core of what we call epigenetics, this layer of instructions that sits right on top of our genetic code.
So let's just make sure we've got that difference locked in. A genetic mutation, that's like a permanent physical change to your computer's hardware. You've literally broken a circuit or fried a chip. It's a fundamental change to the code itself. But DNA methylation, that's like messing with the software settings. The hardware is totally fine, but you're telling it to run different programs, or maybe to shut some down completely. And when it comes to cancer, you can imagine that shutting down the wrong programs can be just as deadly as breaking the hardware.
All right, so if epigenetics is our prime suspect, you might be asking, why has it been so hard to pin down? Well, for years, the crime scene, the tumor itself, was, let's just say, contaminated. It made it almost impossible to isolate our accomplice's fingerprints.
Okay, here's the big problem. When you take a sample from a tumor, you're not getting just pure cancer cells, not even close. It's this messy, jumbled mix of cancer cells, normal cells, immune cells, you name it. It's a crowd. And trying to find the specific epigenetic signal from just the cancer in that mess, it's like trying to hear one person whisper in a sold-out stadium. The signal just gets completely drowned out by all the background noise.
So, to solve this, the researchers came up with this brilliant new forensic tool. It's called CAMDAC, and you can think of it as a computational method that can digitally purify the data. It's a three-step process, really. First, it looks at all the methylation data from that messy tumor sample. Then, and this is the clever part, it uses data from normal cells as a baseline to computationally subtract all that noise. And what are you left with? The pure cancer cell-specific methylation signal. That whisper from the stadium, finally isolated and amplified so we can actually hear what it's saying.
And once they had this clean, uncontaminated data, boy, did they find their first major lead. It turns out the genetic and epigenetic culprits weren't just two separate criminals operating in the same city. No, they were part of the same crew, actively working together.
And this chart right here, it illustrates their findings just perfectly. Okay, so our bodies have these things called tumor suppressor genes. They're basically the security systems that stop cells from going rogue and becoming cancerous. What these researchers found is that cancer uses this two-pronged attack to shut them down. Sometimes it's a genetic hit, a mutation. Other times it's an epigenetic hit. It uses methylation to just flip the off switch. And as you can see, a lot of the time, cancer uses a double hit, hitting the same gene with both methods to make absolutely positively sure it's shut down for good. And the researchers themselves, they spell it out perfectly. They conclude that these two mechanisms, the changes to the genetic hardware and the epigenetic software, they can work side by side in parallel to just completely destroy the function of these crucial security genes. This is not random, it's a coordinated strategy. But then, the investigation uncovered something truly unexpected. A real plot twist in how these systems cooperate. It revealed a new, much more subtle, and frankly, mind-blowing way that genetics and epigenetics talk to each other.
And it all starts with this question. Just think about it. What if the cause and effect wasn't always so direct? What if a genetic change happening in one gene could set off a chain reaction that epigenetically silences a totally different, innocent bystander gene just down the road?
So here's how it works. First, you get what we'll call the loud noise. A genetic event makes an oncogene, that's a gene that promotes cancer, go into overdrive. It gets amplified. It's like someone grabbed a speaker and cranked the volume knob all the way to max, just blasting out signals telling the cell to grow.
Now, the cell isn't stupid. It senses this chaos. and it tries to trigger a silent alarm in response. In a pretty desperate attempt to quiet things down, it starts throwing these methylation tags all over the neighborhood. But here's the problem. This process isn't very precise. So while it's trying to silence that super loud oncogene, it also ends up silencing a bunch of innocent, important genes that just happen to be its neighbors. Talk about collateral damage.
The researchers gave this newly discovered mechanism a name, all-chat. And this, right here, is the crucial plot twist of our story. It's a direct link showing how a genetic change at one spot on a chromosome can trigger an epigenetic change at a completely different spot. It's like action at a distance. It's the ghost in the machine that connects the hardware to the software in a way we never really fully appreciated before.
Okay, so what does all this actually mean for the future? I mean, unmasking the accomplice is a huge step, right? But the real question is, how can we use this new intelligence to fight back and actually change the outcome for patients?
Well, to turn these insights into something practical, the team developed a new metric. It's called the MR/MN ratio, and honestly, you can just think of it as a clue score. It sifts through all the methylation changes on a gene and scores whether those changes are actually functional, meaning are they really silencing the gene, or are they just random noise? If you get a score higher than 1, that's a huge red flag. It points directly to a key epigenetic suspect.
And this table shows that their clue score actually works. It's not just theoretical. By applying this MR/MN ratio, they found several genes where a high score, meaning that gene was being actively silenced by epigenetics, was directly linked to worse survival outcomes for patients. For instance, high methylation on genes like CYP4F2 and MSE, those were powerful predictors of a poorer prognosis. This is the proof. These epigenetic changes aren't just bystanders. They are active players shaping a patient's entire journey.
And that brings us to the final verdict in this whole investigation. DNA methylation is not just a passenger along for the ride. It's not some innocent bystander at the crime scene. It is an active co-conspirator working right alongside genetic mutations and through these really complex mechanisms like all-chat to actively drive the evolution of cancer.
So, this of course leaves us with a really provocative and actually a pretty hopeful question. Now that we can actually read cancer's epigenetic playbook, now that we have tools like CAMDAC and this clue score, we're not just reacting to the disease anymore. We can actually start to predict its next move. And if we can predict its strategy, well maybe, just maybe, we can finally get one step ahead and find a way to checkmate it for good.